And it is also 5 past 4 for me, 5 past 5 for you guys. So if Sylvia, if you're ready, then I think we are good to
I
go.
am. Yeah.
No?
Okay,
Okay.
fantastic. Then I'm handing over to Gautam. Gautam, it is all yours now.
Yes. Thanks. Hello, everyone. Thank you very much for joining us today. Uh, Today, we'll have the pleasure to listen to Silvia De Monte, who for this is one of our first talks actually that involves some perspectives from evolutionary biology, and I was very excited to invite her to share her research. And Sylvia's profile is actually really impressive and highly interdisciplinary. Even very early on from her career, she was always fascinated with how biological populations maintain cooperation despite all the potential conflicts that could occur between their different components. And therefore, she actually chose to pursue a highly interdisciplinary training with heavy focus in physics until her PhD, and then a shift of focus towards biology during her postdoc. So for her PhD, she worked with Eric Mosekilt and Antonio Giorgili on the collective dynamics in populations with microscopic variability at the Technical University of Denmark. And after graduating in 2004, she received the Marie Curie postdoctoral fellowship at the ENS in Paris from 2004 to 7 to work in the ecoevolutionary mathematics group at ENS Paris on models for routing population dynamics in microbial cell physiology. And she did all of this while in parallel pursuing a master's degree in biology. And this really marked her shift towards microbiology. And then from 2014 to 18, she was a lecturer at the same institute. And currently, she is a researcher in the same group, Ecoevolutionary Mathematics at ENS Paris, since 2007. And she also heads her group at the Max Planck Institute for Evolutionary Biology in Germany. The research group is called Dynamics of Microbial Collectives, and the main axes of her research today include ecoevolutionary dynamics of collective function in microbial populations, patterns in community ecology, and artificial selection of communities. Sylvia, thanks again for joining us for this talk. We are very excited to hear you, and the floor is yours.
Thank you very much. So today I'll try to share remotely some of our latest thoughts about how many species communities, thanks to the interactions, self-organize, how this self-organization produces patterns, and the focus of the talk today will be about thinking what kind of patterns can be obtained by simple models where species can be considered to be equivalent in some sense, and what others expectation may we have for communities where species are actually not equivalent. So, sorry. So to start with, I will just say briefly something about microbial communities. As probably many of you know, microbial communities are very rich in number of different species. They are extremely important for a number of functions that go from biogeochemical cycles to the health of hosts, the growth of plants, the microbiota in terms of resistance to pathogens in humans. So we are used to think about this very diverse communities as something that has somehow a common and an emergent function that is sometimes measurable. When people look at communities, not only microbial communities, but in general ecological communities, the observable that traditionally has been gathered is the abundance of different species. So we are usually able to classify what individuals are part of what species. If you think about, a tree community, you can count how many trees are there of different species. And then this is basically a snapshot description of the system. And then from this snapshot description, we want to try and infer something about the ecological and the evolutionary processes that form this community, and also how this community is able or not to ensure to certain functions that can be, I don't know, resistance to pathogens or resilience or just, total biomass. So my talk today is organized in several parts. First of all, I will introduce you with what are the patterns that are observed when one looks at species abundance across multiple samples of microbial communities. And given this kind of observations, I will spend the rest of my time talking about models, which we have used to try and understand these patterns. So first, I will introduce Lotka-Volterra models for species-rich community dynamics. And I will very briefly talk about what happens to models where species are more or less of the same kind, and I will be more precise of this. And this will be the starting point to tell you more about models where interactions are structured. And I will finish by telling you something about ways one can understand the existence of structure in natural communities. So just so that we are all on the same page, here are a couple of slides to tell you how what does one do when we have the number of individuals that belong to every species. Okay, I will hide under the rug all the problem of defining species in microbial communities. So let's start with beetles. In beetles, we know very clearly what is a species. Let's imagine you have beetles, and you know that you can order species in descending order of abundance. So here you would have the most abundant species, here the least abundant species, and then you can just look at the shape of this distribution to determine how diverse is the community. Perhaps you are used to indexes such as the Shannon index or evenness. So there are many, many different indexes that are used to characterize diversity in natural communities. Well, all these indexes are just statistics made starting from this distribution of abundances. Another way to plot the same data is to bin on the y-axis the abundances. So to go and count how many species have abundances inside a certain binning. And this kind of representation, which I will focus on in the following, is called the species abundance distribution. So now in a species abundance distribution, rare species are on the left. So this is the number of beetle species that have, between 1 and 2 individuals. And on the right, you will have the most abundant species. And again, there will be a specific shape to this distribution that people have noticed since a long time, which is like a J shape, an inverse J shape, whereby most species are rare, and there are a few very abundant species. And this is observed basically wherever you look in communities that have a sufficient species abundance. Now, if you think that these distributions are telling you something about the ecological and evolutionary processes a community is undergoing, then you may expect that abundant distributions differ depending on the context. For instance, in different locations or given different natural histories. And this is actually quite difficult to know because very often you just have one snapshot. So we can use models to try and understand what are the underpinnings of the distributions and of the shape. But before going to models, I will show you just an example of microbial abundance distributions that come from a work that was spearheaded by Enrico Sergiacomi, who was a former postdoc with me. and Lucie Zinger, a former colleague here at IBANS, and who is now in Toulouse. So what we did was to analyze the data from the Tara Oceans protist sequences. So Tara Oceans is an expedition that sampled over 100 different locations, nowadays many more, in the world ocean, with 5 size classes. And all this was very deeply sequenced. So this is a very, very powerful kind of database. And we focus specifically on protists. This database allows us to plot one species abundance distribution for every sample. And now if we do this, this is just one example. So for one sample, therefore one size class and one location in the world ocean, you have a species abundance distribution. And then what we did was to find a way to adaptively fit species that are rarest in the community, and this rare species in the end ended up to be 99% of the species that were present in our dataset. And the first thing to notice is that rare species seem to be distributed as with the power law. This is just one example, but this interval you find here is the interval of variation across all the samples of our dataset. And indeed, we found that the decay of rare species seems to be the same everywhere in the ocean. So over the whole data set, there are significant differences between size classes, significant but very small. And if you put everything together, the coefficient of variation of this power law exponent has a coefficient of variation of less than 8%. And since there is very little variation, we also find no really systematic correlation with environmental variables. So we find no gradients or macroecological patterns. So this let us pose, why is this something that we should expect? Usually biologists are interested in things that are different from one place to the others, but luckily physicists are more interested on things that are not changing from one place to the other. And this is what led us to the models. So there are many models that, well, not many, but there are several models that can produce this kind of pattern. The model which we chose eventually to focus on is a model where species are described as interacting with one another, because this seems to be the most appropriate model for an ocean environment. So the equations we use are called the generalized Lotka-Volterra equations. They describe the abundance, here xi, of every species, I, and the per capita growth rate of species I is given by a logistic term that tells you that a species, when it's alone, will saturate at a certain carrying capacity, that is, by simplicity normalized to 1. And it's also influenced by the presence of other species. And this influence on growth rate goes across some coefficients that determine what are the pairwise interactions. And if coefficients are positive, that means that species are competitively interacting. If it is negative, then species are mutualistic, or they, like say, one species has a positive effect on the other. And on top of this, what we add in most of our models is a small immigration term, then it's in principle as small as you want, that has as an effect to avoid the species to go permanently extinct. And I will go back to this assumption. It is quite important to keep diversity alive for long enough, because otherwise species would one by one go extinct. Now, our interpretation is not very refined, but then there are other models that see this as immigration from a meta-community, or mutations or exit from dormancy. You can think about many different phenomena that lead to this rescue of very rare populations. An interesting feature of these equations is that under some general conditions, admits close form or approximate effective descriptions that are basically stochastic differential equations for just one species, which is very practical if you need to solve and derive the shape of a density distribution. And now, with this class of models, we made a couple of studies together with Emil Malmen, a brilliant postdoc who works together with Arne Trausen at the MPI for evolutionary biology in Plön. The first study we did was to consider those equations in the case where each species is, let's say, statistically equivalent to others, in the sense that the interaction matrix alpha ij is randomly chosen according to a given distribution with fixed average and variance. And what we found is that when interactions are strong, which means that if you add another species, you are increasing the total amount of competition that any focal species feels, you would obtain a dynamics that is illustrated here, and that resembles very much what people see in observations. That is, at any time point, the community is dominated by a small number of species, and a vast number of species are very rare. They are so rare that you can't really distinguish them, for instance, on this plot. But the dominant species are not fixed once and for all, as if when the system is at equilibrium, but they turn over with a specific time scale that depends on the mean and variance of the interaction matrix. This kind of dynamics, that is a chaotic dynamics in a very high dimensional space, gives rise to abundance distributions that have a very long power law strand that you can see here, and that we can understand as the interplay between, let's say, the dynamics close to extinction, where species are held close to the immigration rate, and the dynamics, and then they boom, and they stay for a short time close to the carrying capacity before going back. So all this can be described relatively well with a reduced focal species model that allows us to connect this exponent to the statistics of the matrix. And what is interesting is that a similar kind of distribution is also obtained in models that describe explicitly the meta-community structure of the system. And just to give you an idea about what people see when they look at planktonic communities with a very, very high resolution, so let's say at the species level, at the OTU level, they find that there are very intermittent changes in abundance that occur on the time scale of just a few days, which is faster than the time scale of a season or of usual environmental variations in the ocean. So something I forgot to say is that the low-dimensional description that approximates this high-dimensional chaotic dynamics is treating that fluctuations that are due to chaos as a color noise. And we started thinking, well, can we actually generalize this to a broader class of equations? And so Emil came up with the idea of considering a class of equations where, so this n is what was x before, so it's whether the per capita growth rate of species I is actually stochastic, and the growth rate is a color noise term. And then we also have interactions, which we have chosen here for simplicity to be the same with all species different from your own species, and stronger within a species. And this is this term. And what is interesting is that in this system, again, there is this immigration term that avoids species to go extinct forever. So in this set of equations, it turns out that the long-term steady state is again something that resembles a chaotic turnover. Here is a stochastic equation, is not a deterministic one, but in certain conditions, this resembles very much what we observed for the deterministic case. And what is interesting is that depending on just two aggregate effective parameters, so that combine the 7 parameters that are present in these equations, one can go from different shapes of the abundance distribution. So this shape that can generally be well fit by a generalized inverse Gaussian distribution resembles very much what one finds for plankton. However, under conditions where the immigration rate is higher and the self-regulation of any species is also higher, then you would get unimodal distributions. And these distributions can be fit well with log normal distributions that have been used a long time to describe the SAD of macroorganisms. But you also get some skewed distributions like gamma-like distributions that have been used to describe microbial communities in the gut. So it seems that there is not one best, just one single solution for what is the best distribution. It will depend on some features that are actually less detailed than deciding if a model is deterministic or stochastic, if it is neutral or if it is niche, but have to do more to the way species avoid getting extinct and how they interact with one another when they are at very high population density. So this is to say that these simple models where species are considered basically to be the same do quite well in explaining a number of patterns that are commonly found in microbial populations. And these kind of patterns can be obtained also by these effective low-dimensional descriptions, which is a good thing because we can do calculations on this. However, if you think about the traditional models in ecology, there have been a number of other models that are low-dimensional and that are not at all of the same nature of these models that rely on having many, many different species and doing some kind of statistical averaging. And these models are, for instance, NPZ models, so resource consumers models, models where there is a functional description of what actually a certain set of species does in a community. And then these models sort of forget the identity of species within the same functional class, and they say, okay, what is important is just that there is, some autotroph and some heterotroph and some predators. So the question I would like to talk about is when and how do predictions of this kind of different low-dimensional models differ, when are they the same? And in the final part of my talk, I would like to discuss briefly what shapes disorder and structure in these species interactions. So the first part, the first project I'll be talking about has been basically conducted by Juan Giral Martinez, whom I have co-directed with Mathieu Barbier, and at the beginning also with Jeff Arnoldi. If you're curious to know the details, this is published. And the idea was to again start from a Lotka-Volterra equation, but now define these interactions in a way that reflected some ecological knowledge we may have on the biological system. So typically people take alpha ij's as random, and since people use results from statistical physics, they choose the statistics of this matrix to scale with the number of species, which means that in this case, if you add more and more species, you keep the average competition constant. So we will do the same, but we want to add to this some structure. And this structure doesn't need to be weak. So it is possible that species within functional groups, they interact weakly between them, but they interact very strongly with another functional group. And this is something that people observe actually in natural communities whenever there are functional groups. And now the question is, how do we define the structure? A very general way to define structure is to decide that there is a number, typically a small number of functions, here, for instance, F1 and F2, and these functions can receive contributions from every species of the community, and this is measured by what we call the impact rates, or what ecologists call the impact rates. And at the same time, these functions can feedback on each of the species through some other coefficients that are called the sensitivity traits. And in general, the cases I will consider will have a number of functions that is much smaller than the number of species. This kind of framework allows us to encompass different functions. For instance, you could imagine there are functional groups, so that every species who is within one functional group has basically the same impact on the combined abundance of the functional group, and this combined abundance will be the factor that influences all the species of another functional group. Or you could imagine the existence of public goods or resources where species individually contribute and respond to. Or you could imagine that these traits, I and S, are determined by some kind of phylogenetic structure. So for instance, by their phylogenetic diversity, and then you would obtain a structure matrix that has this fractal structure. But for the sake of simplicity, in the rest of the talk, I will focus on this kind of structure. So a case where there are just a small number of different functional groups. So if one applies the same approaches that people use for the usual disordered Lotka-Volterra equations, that is the cavity method or dynamic and Mean-field theory, one can obtain that also in this case, we can reduce the dimensionality, the effective dimensionality of the model, but this time, considering one single equation is not enough. We need to consider that the equation for every species is coupled to a small number of macroscopic, or if you want, mesoscopic variables that are the functions. The interesting thing is that the equations for the function are actually only dependent on the functions themselves plus a correlation, a time correlation. So NF plus 1 macroscopic degrees of freedom are basically accounting for the behavior of the in principle infinitely dimensional equation. And interestingly, at equilibrium, these different degrees of freedom are coupled by some self-consistent relations that depend on a parameter that is the combination of the spread in heterogeneity, so the standard deviation of heterogeneity, and the correlation at equilibrium. So let me tell you just a couple of implications of this. So the first implication is that if you think about starting from a community that has, for instance, 4 functional groups, and these functional groups, each of them has a certain equilibrium value, so that when species that belong to the same functional group are strictly identical, then all these species will have the same abundance. And now you can think what happens if I start spreading heterogeneity. So I go from gamma equal to 0 to positive gamma. Well, the value here, the model value doesn't change that much. It changes a bit, but mostly I am spreading the distribution, and the species abundance distribution will be the envelope of all these distributions, which means that some species that belong to a functional group that would go extinct if it was not for heterogeneity would instead survive, and vice versa, some species of groups that would have survived would go extinct. So there is an effect on diversity, but this effect is somewhat trivial because all these distributions are Gaussian distributions. On the other hand, this indicates that in principle, if there are some very strong functional groups, we should be able to see this in species abundance distributions. And if we don't see multimodal species abundance distributions, perhaps this is the indication of the fact that there is no very strong relation, like trophic relation, in the data set we are examining. So more interesting observations come when one looks at the existence of a low-dimensional dynamics to start with. So this low-dimensional dynamics, as I was mentioning before, has been looked at in many, many different models, and people were particularly interested in out-of-equilibrium dynamics, where you have oscillations that look similar to what one observes in many time series. So let's imagine we start from such a model. And now we can ask, is such dynamic destroyed when I take into account that species within the functional group are not all the same, and actually I start spreading this sigma, increasing this sigma, and making species more and more dissimilar to one another? To give you an example, imagine you start with a matrix like this, where the parameter k determines what is the influence of one functional group on another one. And these functional groups obey a rock, paper, scissors kind of relation, which makes so that when these are just 3 species, then the dynamics would be cyclic and oscillatory. And if you take, as many species as you want, but within each of these classes, species are identical, not surprisingly, the average abundance will have exactly the same behavior as the case where you don't have diversity. Now, if you start increasing this order, what happens is that at the beginning, species within a functional group keep oscillating in phase. I mean, their individual behavior will change. Their individual behavior will change, but on average, you will still see macroscopic oscillations of the functional groups. But since species are more and more diverse with respect to one another, the amplitude of the oscillations will decrease. But this amplitude will decrease so much that for some intermediate values of disorder, you will miss completely oscillations. So species will go at equilibrium, even though if you took out triplets of species, one belonging to each functional group, these three species would oscillate. So this means that you have stabilized the dynamics by increasing heterogeneity, which is something that is contrary to the prediction in the seminal work by Robert May, who said, actually, if we increase diversity, we should expect stability to be lost. However, if you are here and you keep spreading disorder, then you will eventually end up in a case that corresponds to May's transition, where species will start oscillating independently from one another, and on average, these oscillations will be stable, even though every species oscillates. So these kind of transitions can be explained by looking at the Jacobian matrix of the system that is composed of a bulk of high density of eigenvalues that basically derive from the random part of the interaction matrix, plus some outlier eigenvalues. And these outlier eigenvalues undergo a bifurcation in going from here to here, whereas in going from here to here, it is the bulk who crosses the imaginary axis. And this can be understood by noticing that the Jacobian matrix of this system can be reduced to a pseudo-Jacobian matrix that has the same eigenvalues. So this is the full matrix. This is the pseudo-Jacobian, and the outlier eigenvalues of the pseudo-Jacobian are the same as of the original Jacobian matrix. And interestingly, they depend on the abundance at equilibrium of the different species. This is somehow telling you that differences in equilibrium abundance reflect in different time scales that are present in the population. And when you are coupling many different time scales, at a certain point, the system can stop oscillating in something that is known in synchronization theory as oscillation death. So this means that it is possible that if you have species that are interacting, that belong to functional groups that interact strongly, but the species are very diverse with respect to one another, you may not see the expected fluctuations that you would obtain if you only describe the interaction between the functional groups. Okay, since I would like to have to go very quickly through the last part, I'll probably just let you read in a second time this conclusions. And I want just to point out that here we have imposed a structure, and the question that one can ask is, when does such a structure emerge? When do we expect to see it? And in order to answer this very partially, this question, because it's a very vast question, I would like to tell you something about some work done with Jules Freboul and Giulio Biroli that is about what happens to a random matrix when we select a given function that this community is providing. And this happens through community level selection that models, if you want, something that is thought to happen, for instance, to the microbiota. So you have a structure of reproduction of the community whereby the community is judged somehow according to its function, for instance, relative to the host. and then can be vertically transmitted. So in the lab, this can be realized rather simply. You take, simply in quotes, you take many different communities, you let them grow for some time, and then you measure a certain function of interest. For instance, imagine community biomass. So you have, let's say, newborn communities that grow into adult communities. You apply selection, you only select the best performers, and then by some means you reproduce and or mutate these communities, and you go on over and again, and the expectation is that in this way you should increase the community function. Okay, when people have tried to do this, it doesn't always work. But with, here is a couple of papers if you're curious about this. But what we thought of doing is that we could try to model this and try to understand in which conditions do you expect this artificial selection to be effective. So the model goes like this. So you start with a community, Lotka-Volterra, exactly as I described before. And now you let this reach equilibrium. At the end of this growth phase, you judge communities according to a certain target function. For simplicity, I will take like total biomass. Then we pick the best community and we'll duplicate it in a way that we will introduce some variation in the matrix of interactions and let it grow again, and so on and so forth. So to explain this model would take too long, so I will briefly go to the conclusions. I just say that, so the ecology, as I said, is Lotka-Volterra. We start from an ancestral community that is at equilibrium and that has a relatively low total biomass. And then we expect that evolution should push up this gradient towards higher and higher biomass. Mutations, if you want, we can discuss this later, are applied at the level of the whole community. And this is what allows us to have unbiased mutations upon which selection can act. And selection will try to maximize this product of the equilibrium abundance times the target function, that is the vector 1 in the case of total biomass. Okay, so this is just to illustrate that you start from a random matrix, then you will have several matrices, and of all these matrices you will just select one and go on. And the magnitude of the mutations will be given by a certain parameter. You will have certain mean and variance of the initial distribution of the initial interaction matrix, and we will distribute also carrying capacities of the various species. Now, if you do this selection process, perhaps unsurprisingly, you will end up increasing biomass. This is something that one finds in many other also works of artificial selection. Now, what happens to the matrix? If you start with a random matrix, you see that the final matrix doesn't look completely random, and it definitely looks more mutualistic than it was at the beginning. This actually happens due to a change in the spectrum of the matrix that consists in the emergence of an isolated eigenvalue that acts like a global mutualistic interaction term. So species become more mutualistic. We can describe this mathematically, but I don't have time to go into the details. What is important to know here is that not all species react in the same way to this selective pressure. So there will be some species, notably those that initially are more abundant, that will become more mutualistic. And the structure that is imprinted by adding selection at the level of the communities can be somehow guessed through measuring only community level parameters, such as the average interaction strength, its variation, and the composition of the community. So this let us believe that perhaps it would be possible to infer when the transition between totally random matrix and a matrix that has rank 1 structure happens during the course of artificial selection. So just jumping to the conclusions, applying community level selection to complex interacting ecosystems generically imposes a structure on species interactions. And this structure, something I didn't have the time to explain in the details, but depends both on the ancestral community, the structure of the ancestral community, and on the target selection, which means that not all selection targets will match the initial community, which might explain why these selection experiments sometimes work and sometimes they don't. So the structuring process has a statistical signature that is the same basically for all evolutionary trajectories, but that produces communities that are not identical. So we are now working on trying to understand the multiplicity of these solutions. And in particular, we are interested on the history dependence of what kind of community you will end up having evolved or selected. And with this, I thank you all my collaborators, including some colleagues with whom I had great fun with an art and science project that is representing chaos in plankton communities. And thank you very much for your attention.
Yeah, fantastic. Thanks very much for the fascinating talk. It was really interesting.
Yeah,
I've
thank you.
got already loads of questions, but maybe first the floor to the audience. Are there any questions in the room?
You could just raise your hand, unmute yourself to ask the question, or you can also write it in the chat, or I think there's a Q&A. We will read it out to Silvia if you'd like.
Yeah, maybe I can start with one for people to get a bit of more momentum. So this is maybe less of a question, more trying to make some ideas meet that I haven't met before, basically. So you mentioned in the beginning that for populations, microbial populations, very often you see this log-log distribution where you have a few very abundant species. Most of them are like in the low numbers, and that here, when you have now a specific interaction coefficient between them, they show well, they change from an oscillating behavior to a flat to a chaotic change of their abundance numbers. Does that somehow summarize what you were saying?
Yeah, so there are different chaotic regimes. I mean, there are various possibilities, but the distributions of abundances are actually on when you are at equilibrium. The distributions that are predicted by these models are typically these Gaussian distributions or truncated Gaussian distributions that do not correspond to what we observe in nature.
Right.
So if you want to get this kind of distributions at equilibrium, you need to basically input them in the way you define the parameters. So the parameters, the traits, need themselves to have a power law, and that would reflect on having a power law of your distributions. And this is what people do often in modeling. They say, okay, I make this hypothesis that things are distributed somehow, I don't know, log normal if they want to obtain a log normal distribution. So to me, that is not really solving the problem of how do you get this power laws, but it might be a reason, and there are many reasons to believe that certain traits may be distributed like a power law, because, for instance, they may be related to size, and size in, I don't know, in marine ecosystems is distributed as well as a power law. Mm-hmm. So it is possible that that is just the explanation, and then, you have a system at equilibrium with traits that are distributed as a power law that give rise to power law density distributions. But the other possibility, and this seems to be the case in many planktonic ecosystems, is that actually the system is out of equilibrium. And when it is out of equilibrium, then you get these power law distributions with varying with varying slope, and then depending on
one, it's not coming at all, and the other is too loud.
Sajid?
Excuse me?
I think Sajid is at the moment not muted.
It sounded like they were asking a question too.
Right.
But anyway, it's okay. I think
here.
There, sorry to interrupt. It's Marco has a question. They have their hand raised.
Yes,
please go ahead.
Ciao, Silvia. Thanks for the talk. It was really nice, as always. And just wanted to ask about the extinction. So you keep the number of species up as you optimize the matrix, if I understand correctly. And you get a certain hopping within high dimensional space.
Yeah.
But if you start to allow things to become extinct, then you will lower the dimensionality, and then would that then move more towards what you would expect from like the coarse grain functional groups sort of behavior, more oscillatory, or what would the dynamics be?
No, in general, what you have is that you may go through an oscillatory phase. I don't think we have checked this, but in general, so first of all, if you don't have, in the Lotka-Volterra case, if you don't have immigration, you go into this like glassy phase where you jump from one equilibrium to another one, but it takes longer and longer to jump from one to the other one. So in the end, if you put a finite threshold of extinction, as one should, to be consistent with the fact that the number of individuals is not continuum, then you will get extinctions one after the other. And then as species get extinct, they will eventually go to an equilibrium, because if you don't have high dimension, you tend not to have this oscillatory dynamics.
Okay.
So it's, let's say, there are some phenomena that seem to be related to the existence of high diversity, and the moment in which you reduce drastically the number of species, then you lose them.
Okay, thanks.
You're welcome.
Jonas, please go ahead.
Okay.
Yeah, hi. So this is very much not my field. So I'm
go ahead.
I'm trying to understand like a bit the timescales of the underlying mechanisms from these dynamics. So thinking of like ecological mechanisms in terms of growth and nutrient availability and so forth, versus evolution versus plasticity. Like, how do these things come together in this?
Yeah, so basically except the last part of my talk, we basically ignore evolution. In general, we ignore plasticity, even though we are now working on models where, you can expand the scope of your description, and then say, okay, each species has a dormant phase, for instance, and then you have a certain probability of switching to a dormant phase, and then you can study, this kind of system where you have the vegetative and dormant cells, which again gives rise, to variations on the theme of these different regimes. That I have basically described. I have described to you the most common regimes that one encounters, but the effect of different parameters on these regimes changes depending on the exact system you are modeling. So this is for as far as one can go for physiological variation, even though I think is super important, and I'm starting to think that actually this description of diversity within a functional group may somehow encompass physiological diversity, even, I don't know, within one species, or I mean, we've done some simulations when actually you have many species. Actually, the first model I presented. And then you let each of these species have many different strains, and then you get basically the same result. You start with something that is chaotic in high dimension, and eventually you stabilize it. In that case, you get cycles, and it's something we still don't understand very well. So it's definitely, I think, a biologically very relevant question, this of timescales. But here we sort of separate out the timescales. Same for evolution. We don't let evolution occur at the same rate, or let's say on the same timescale of ecology. It's something we are doing now. We are not the only ones. There are several people like Daniel Fisher in Stanford who are thinking actually on how to integrate ecology and evolution in this kind of models.
Any other questions from the audience? Okay, I have a couple of questions. I'll ask one of them. So first, actually somewhat related to what Jonas was just saying. When you were going through the last part of the talk, you said that, for example, in the case you have the influence of evolution, it would lead to higher and higher biomass, which I'm assuming I'm sorry if I'm wrong. I don't know much about this field. So the assumption is that it's natural selection primarily, and therefore reproductive fitness, and hence higher biomass, right? But what about in the cases of these microbial communities? If you have coevolution in these same timescales, wouldn't that change this?
That's sure. So something I didn't have the time to explain is that we chose these mutations to happen at the level of the community because we were mostly interested in the effect of selection at the level of the community, which means that species basically mutate all at the same time. This allows us to have like a reference model in a case where we don't apply selection at the level of the communities. We observe neutral drift of the function we are selecting. So then we can evaluate the effect of selection with respect to neutral drift. But it is correct what you're saying, that in general you will have evolution within one generation, one community generation. And this is something we are starting to do now with a PhD student who is working with me. We are doing more or less the same kind of scheme that Daniel Fisher has done. So they have devised basically an adaptive dynamics framework to describe what happens within a community. But then we want to have at the same time selection acting within a community and between communities. And what we expect is that there may be cases where the two align, but in most cases they will go in opposite directions. And this may also be a source of inefficiency in community level selection.
Okay. Okay. Interesting. Thank you. I see. Okay, it's not a question. So Marco Polin says snazzy animation, by the way. I'm assuming for the animation on the slide, yes.
Yes. I have more on this. They even dance this. It's crazy. Interacting with artists is really interesting.
Nice. I have one more question if there are no other questions from the audience. Okay, seems to be the case. So this is also just a quick one. I was always curious about the inference of the matrix structure. I'm assuming you were talking about the alpha matrix, right?
Yeah.
So I saw earlier in the inference methods there was the functional one, which is sort of the direction that was taken initially, and the phylogenetic one, which is the history, right? But then is there any way that's just by doing experimental evolution studies on very simple communities that you can infer such a matrix? Is that even possible?
That's what some people try to do right now. So usually it's not evolutionary, it's ecological studies, because these experiments already at the ecological timescale are really very demanding, because people assemble communities in the lab, and then they follow this for, I don't know, a couple of weeks, measuring the abundances of different species. And so this is one, so there are some methods to try to infer this matrix in this way. Other people measure pairwise interactions, by making all possible pairs of species, or all possible triplets, or sometimes a subset of couples and triplets. And so there's a lot of work that is being done in this moment. I am, for instance, collaborating with Dani Amor, who works on this kind of communities assembled in the lab. And the thing we are thinking of, and we are not the only ones, but is how to describe well the functional landscape that is generated by this different combination of communities. And there's a lot of work in that direction now, also using machine learning, using inference to try and guess what is the functional landscape and how much does it depend on the details of the community composition rather than on some coarse-grained features of the community.
Okay, nice. Okay, thank you. Marco had raised their hand again.
Yes, and then I lowered and then I raised it again because I'm not sure whether you have already answered the question that I wanted to ask, which is in your matrix of interactions, right? You're gonna have I think might be possible to understand some of these interactions as predation or being a prey, or and so maybe you can have a loop of this praise that praise this, sort of like your rock, paper, scissors example that you had before. And I was trying to link this to what you said about the emergence of cooperation, right?
Yeah.
And so in terms of stability of the community, I was wondering whether having something that is, for example, a mix between cooperation and maybe competition or different trophic levels that emerge from your optimization is something that makes gives different qualities, let's say, different attributes to the community. Is there something that emerges, or can we understand it like that?
So this is an excellent question. I don't think I have really a ready answer. So the the example I presented of chaotic turnover, that's a matrix that doesn't have a precise symmetry, so the correlation is 0. So it contains both competitive and facilitative terms. And the same for the evolved matrix, the matrix that we let evolve. So it is true that a part of the interactions become more mutualistic. It's not easy to interpret this as, in terms of trophic levels. Here we are thinking more about what could be, mixotrophy or, these interactions can change in nature. They can go from competitive to mutualistic. And this is also something that may not be really that easily realizable in nature. So I guess that a further step to understand better something related to your question is to actually, instead of considering this Lotka-Volterra models that are very implicit, is to go to consumer resource models that have other kind of limitations, but at least they are explicit in the way actually the interactions occur. And there it would be easier to ask that kind of question.
Thanks.
Thank you.
I think that's the end of all the questions. I will stop the recording